a little bit differently here. Um, like I told you in class, that I thought it would be great if I go back and do some screencasts of your PowerPoint presentations to kind of help make some of these concepts make sense and to go into a little bit more detail about some of these concepts that our class time just doesn't allow for us. And of course, if you have any questions, please feel free to email me. Or if you want to wait till class, make sure you write down the questions you have, and we can address those questions when we meet. Okay? So we're going to kind of pick up where we left off. Okay? So we're in chapter 4 of the neuroscience. And so far, we have been talking about some of the research techniques and methods that neuroscientists typically use when we're examining the brain. And when we examine the brain, we are examining its function, the parts that's necessary for functioning, where there may be dysfunctioning, and areas that can help improve research in this particular area. So far we've talked about some of the structuring and imaging techniques that's typically used, such as the Golgi stain, the myelin stain, the Niesel stain, as well as autoradiography and immunocytochemistry. Okay, so we start talking about some of the different types of microscopes that may be used when we are examining some of the fine details of the brain and functioning. So last time we left off, we're talking about the light microscopes, okay? And with these light microscopes, these are going to help us to look at cell bodies. So we're looking at structure here. We're looking at things that's more so on a larger structure, like the dendrites as well as the axons. And though ideally, yes, we can look at more in-depth detail of some of these larger structures, we have a limitation here. And one of those limitations is due to the nature of the light. This is a light microscope. So the lighting actually um, kind of has limited capability, if that makes sense. So because of the nature of the light from that kind of microscopes, it limits how much of depth we can look at these particular structures, if that makes sense. Okay. Then we have electron microscopes. Okay. So with electron microscopes, what's going to be the main thing about this is it passes beams of electrons through a thin slice of tissue. So and it magnifies up to about 250,000 times and can distinguish features such as small as a few hundred millionths of a centimeter. Okay. So that's kind of one of the good things about this, and that this can actually detect a little bit more through a beam of light. And yes, it's going to be on a higher resolution, so you can be able to magnify these images. So think of this as like a 3D kind of thing. Okay. And of course, with the electron microscopes, there are two types. Okay. Which, let me go back for a moment. So if you have your pen and paper, you will need to note that there are two types of electron microscopes. There are transmission electron microscopes, and then there are scanning electron microscopes. Okay. So what a transmission electron microscope is that it works by, of course, passing the beam, just as we saw before, um, a beam of electrons through a thin slice of tissue. All right. With a scanning electron microscope, the beams of the electrons induces the specimen to emit electrons itself, and these are captured just as the conventional microscope collects reflected light. Okay, so that was kind of a lot to kind of break down and tell you. So the easiest way to remember is that with the transmission electron microscope, um, it passes through the electrons at different degrees. So it can produce the image on top of the film or the video screen. Okay? Give you a moment to process that. Now, with the transmission electron, yes, we are able to see different degrees of the image through these beams of light. What a scanning electron microscope allows us to do is that it allows us to magnify the image so much so that we can see things such as the synaptic vessels in the axon terminal. Okay, so transmission electron microscopes passes a beam of electrons through the thin slice of tissue, and we're able to look at different degrees of these electrons, so and it produces the image that we're able to see just like this picture here. The scanning electron microscopes allow us to be able to look at a much more deeper detail and more in-depth detail of this image, such as the synaptic vessels that is on the axon terminal. And again, as you can see here, this is what we mean by the 3D effect. So we can remember that scanning electron 3D, okay? Now, let's get into some of the research techniques that you probably may be familiar with. And if those of you have ever seen any type of research studies or sleep studies, you may have seen this image before right here. So this is what's referred to as an EEG, okay? And let me back up just for a moment. We've already said that staining, one research method is staining and imaging neurons. The second research method so far we talked about is light and electron microscopy. So now we're moving on to different types of ways that we measure brain activity through manipulation, okay? And this is one of them. So the EEG, or the electroencephalogram, which hence why we say EEG in our profession, that's a pretty long word to say. Nonetheless, it's one of the most useful types of research methods used in terms of neuroscience and imaging techniques. So, what the EEG allows us to do is that it allows us to detect any type of electrical activity that's going on amongst all the neurons by using these little electrodes here. You see those? Those are the electrodes. What that allows us to do is that it allows us to detect different sorts of brain activity that may be going on that's just not quite observable to the eye. Okay? And these types of what you think is maybe a scribble, or for some people looks like a signature. This is the output of these types of techniques. So this is the recording of the activity. What's interesting here is that when in our profession, when we do sleep studies, we study what we call REM sleep and different stages of REM sleep. And so what the EEG allows us to do is detect, based on the type of brain waves here, what stage of sleep that individual might be in. Okay? Um, and the EEG is really great to use. One of its major advantages is that it has good temporal resolution. And when we're talking about temporal, we're talking about time. So we're catching things in real time. It's pretty much what this is saying. Okay? However, one of the downsides of having good timing with our resolution is poor spatial resolution. And so when we're talking about spatial, we're talking about its ability to detect specifically where is the activity or the signal coming from. So we're not really able to see a good picture of that. But nonetheless, it's still great in that it helps us to see any changes in arousal states. This is also used for individuals who, I know in my experience before, we've dealt with coherence studies where we're looking at heart rate variability. And I know with the heart, that's a little bit different, but we can also detect brainwave activity as well when a person shifts from a state of coherence, which is a state of calmness and peace of mind, from getting to a state of arousal like anxiety. Okay. So remember, with the EEG, it allows us to detect brain activity amongst the neurons by using electrodes. One of the good features about the EEG is that it allows us to catch these brainwave activities in real time. However, it doesn't give us quite a clear picture of where it's coming from and what it would look like. Nonetheless, it is helpful in how we're able to see when a person changes from different arousal states, such as stress to calmness. Okay? give you a little minute to process that because I know that was a bit much. Okay, so with the EEG, we have something that's called event related potential. Okay, and so with the event related potential, what this does is that it's able to produce over several presentations of the stimulus. Okay, in that one single trial. Okay, so let me think of an example to help this make a little bit more sense to you. So with event related potential, um, it allows us to see different presentations. So remember this right here, what we saw before? These are various presentations, right? Okay. So what the computer does is it averages the EEG over several stimulus presentations, okay? So let's say a stimulus may be a noise in this case. What it's able to do is take an average of several times we may have presented that noise to that particular person and allows us to watch the change in activity at the same time. Does that make sense? And if not, it's okay. You can write a question and we can talk about it more in class. And also, it cancels out the noise of the brain's other activity. And yes, our brains it makes noises on the inside, just like our stomachs when we have our digestive system. It works the same way. So it's great in that it helps cancel out the noise so we're able to focus more so on the activity than the noise drowning out our ability to capture that activity. Okay. So now we're getting into another technique, which is through stimulation, recording, and stereotaxic techniques. Okay, so right here, a stereotaxic atlas is pretty much something that tells us the location of some of the key structures of our brains based on images. You would kind of sort of see this like in your textbook at times, where they may show you an image of where these various parts are located and what they may look like. Depending on if you're doing research, certain research programs may let you look at a sample of the specimen that they've collected and the atlas that they've created of where these particular brain structures may be located. Many neuroscience scientists rely heavily on these types of atlas because when you're doing brain surgery or you're thinking about research rather, you kind of have to understand where these particular images are located and what sections of the brain are they located in because some structures can be in between two lobes of the brain, but you'll still be required to know specifically where these parts are. We can't just go drilling on the brain. I'm just going to find the golden treasure wherever I randomly find it. It doesn't quite work that way. You got to have a map before you can do anything else. And that makes sense. So it does allow us to kind of get a more precise positioning of the electrode should we decide to use the EEG and where to place it on the brain. Okay. So it helps to determine where we're going to pretty much locate a probe or where we're going to place a probe for a study. Now, it's not easy coming up with an atlas, and it's not something that people just randomly draw on their own, but rather how they construct these kinds of atlas is that they have they have to get a large number of brains that are thinly sliced into very thin coronal sections. So that's kind of sliced very, kind of thinly. And the drawings, kind of help them prepare to show where the average location is of those structures. So you need a real brain in order to make these types of atlas to know where these structures are. Now here in this example, as you see here, Of course, and you're going to find with many of these studies in neuroscience, they're going to be dealt with animals. Animals are going to be the primary use because it is unethical to use a human being for these kinds of studies. And so here, what they show is a stereotaxic instrument for rats. So the instrument secures the rat, which they use an aesthetic. So they're pretty much numbed right here. They're numb, so they're not really feeling this at all. And the investigators then insert a probe through a small hole drilled in the skull, which you'll see here. See, and at the precise location and depth that's specified by the atlas. So they have an atlas for a rat, so they know where to place this at and how far to go. And here is the actual stereotaxic instrument, which is used. As you see here, this is the rat. And of course, they're using aesthetic, so he's pretty much numbed. He doesn't feel any of this. And you can see where the hole is going to be drilled. So you have the atlas that shows you where to go. Here is the equipment to do so. Okay. Looks kind of vicious, doesn't it? Kind of scary idea of a little animal sitting there with a hole being drilled in his head, but that's what the rats were for before. So moving right along. And we kind of talked about this a little bit more. If you want, you can refer to your textbook, and it kind of gives you a breakdown of each one of the parts that's used to do so. And when we get to class, we're going to talk a little bit more about lesions and damages to tissue, a little bit more, as well as some human subjects voluntarily used for some of these studies. But for the sake of time, we're going to keep moving on. Okay, so now we're going to get into some brain imaging techniques that you might find very familiar that you've heard of before. So on the board, I pretty much drew a layout for you on the easiest way you can remember some of these structures, okay? So the easiest way to remember this, and if you have a pen and paper, I suggest you write this down. Um, we have to think of these particular brain imaging techniques based on structure and function, okay? If we're looking at brain imaging techniques that's based on structures, it's going to do just that. It's going to help us locate where the lesion may be. It may help us locate where the tumor may be. It may help us locate where a particular part of the brain may be dead already. It helps to locate the structures of the brain, okay? When we're talking about functioning, that's one of the disadvantages of the imaging techniques that focus on structure is that there are imaging techniques that help us locate functioning, whether there's a lack of neuronal firing of our neurons, or if a certain portion of our brain isn't responding or it isn't stimulated. Therefore, we're not able to have a reflex or use our five senses and things of that nature. Okay, so that's the easiest way. So with imaging techniques that allows function, it shows us the structure, but it also allows us to see the functioning of the structure too. Okay, so one of the first imaging technique we're going to go into is a computed tomography, or really known as a CAT scan. Okay. This is typically what's done if you have a person with a brain injury who may be coming into um, the emergency room, or a person who's just recently had a stroke. Well, immediately they probably may do a CAT scan on that individual as a standard procedure. A person who may be experiencing chronic migraines to the point where they're blanking in and out, that may be the first standard form of procedure when they come there is to have a CAT scan done for them. And so, what the CAT scan allows you to do is that it produces x rays from different angles. And so, using a series of like two dimensional sections or slices, rather, they can go through these areas as if they were 3D images, okay? Now, the way they do that is that dye is kind of injected in the blood. So remember, the dye is to help locate where these particular structures are, depending on what brought the person in, okay? It also will show you the density of the blood vessels, which your brain does have blood vessels there. And it gives real graphic 3D X-rays of these images through horizontal sections of the brain. Okay, so a CAT scan or CT scan it's an X-ray, and we know what X-rays are. X-rays help us look at the structure. If you ever broken your arm or your foot or your wrist or your ankle, you've had an X-ray done to show where it may be broken at. That's a structure. Same thing with the CAT scan. Only difference is that with your brain, this dye is injected into your blood. Therefore, it allows us to locate the structures that we may be interested in, because it's not just going to show up on its own. We have to inject something so that way we can locate it. Okay? And with the CT scan, it uses two-dimensional horizontal sections so that we can use them almost as if there were 3D images to locate these images or these structures and see just what's going on here or what it looks like. Okay? Now, just like with anything else, there is a disadvantage of the CT scan, okay? And one of the major drawbacks for the CT scan is that, well, previously the CT scan tended to be very slow in terms of producing the images. So, in other words, you ever was waiting for a text message from a person, they sent you a picture, and you're sitting there waiting for it, and because maybe you're in a bad area, it's taking all day for you to get the picture. Well, that was kind of some of the same similar issues with the CT scans back then. Now, of course, newer models have updated that, and these images are supposed to be much quicker. I would like to think so, being that they are using a standard form procedure for people who have strokes. Nonetheless, it's still it's something that is carefully monitored. Okay. And as you can see here, this is what happens. I myself had to have a CT scan back in 2013 when I found out I had migraines. And you see this machine right here? This was the very machine I had to go into. Now, if you look here, well, of course, this is the X-ray source, which is where you see in this tunnel. Some people, some people have anxiety about this particular type of process, but I reassure you, it's okay. Okay. So I'm gonna get back to this. Oh, what am I doing here? Okay. I don't know what's going on here. We're gonna do that. All right. So as we see here, this is that image that we were talking about. So this is from a horizontal perspective, and if we look closely right here, that is a tumor. So what the CT scan allows you to do is locate where the tumor is, and this is a structure. See? Now, the next one is the magnetic resonance imaging, or we know it to be as an MRI. Okay, we've all heard of that before. Okay, so this is another type of imaging technique that allows us to locate structures. The difference here is that it uses radio frequency waves. Um, and this is more about science when we're talking about it being emitted in the nuclei of hydrogen atoms when they're subjected to a strong magnetic field. I'm not going to go into that too much because that can confuse you. The main thing to remember about an MRI is that just like the CT scan, this is also a scan, but um rather than using the dye to go into the brain tissues, this operates off of radio frequency waves. Okay? So, as we see here, this is an example of, again, we're looking for the structure, all right? And if we're looking at the fibers, which it shows all the structures here, the fiber structures is connecting your four lobes, the frontal, parietal, temporal, and occipital lobe areas. And the colors were added to provide some of the dimension so you can know where these particular structures are. So through the radioactivity, we're able to still locate where it is, which is here. Does that make I hope this makes sense for some of you. Again, we can talk about the images more in depth when we get to class. One of the major advantages about the MRI is that is very fast, especially in detecting some of those small images like here. You see here, and as well as these fibers. It's very quick at detecting that compared to the CT scan. Okay. And it's more versatile. So you're really not limited here. And they're very inexpensive. MRIs, believe it or not, can compared to other brain imaging techniques tend to be less expensive and even portable. So you ask yourself your question that what are the advantages of a CT scan and an MRI compared to other brain imaging techniques? That's a question you want to keep asking yourself to make sure you're understanding conceptually what these type of techniques are. And of course, again, just reiterating that a CT scan and an MRI have some of the most greatest capability for detecting tumors, especially if we're looking to see if there's a relationship with possible brain damage here. And with behavioral symptoms. So, for instance, when we're talking about a person with a stroke, for example, and they're coming in, and we don't know if maybe a tumor could have set that stroke off. And with the stroke, typically a person experiences the face drooping, the slurred speech, and numbness in one side of the body. So those are behavioral symptoms to something that's neurological that the CT scan and the MRIs are able to capture and detect. However, although they're great in catching these tumors, they do lack the ability to detect the changing in brain activity. For example, that EEG, remember how that allows us to detect changes in arousal states and all of that? Well, the CT scan and the MRI are not able to do that. So now we get into thinking about more functional types of brain imaging techniques when we're interested in that. So here we have what is called the positron emission tomography or the PET scan. Okay. And so with the PET scan, then, kind of similar to the MRI in that it injects radioactive substances into the bloodstream. So these radioactive substances it goes straight to those parts that we're interested in looking at. That's just like, let's say if you were um let's say you were drawing a map and you need to color code those maps to know what they are. Well, that's just what the radioactive substance is, okay? It's injected into the bloodstream and it goes to those most active parts of the brain. So remember, right now we're talking about functioning here now, okay? So when we're talking about active parts, we're talking about functioning. We're talking about brain activity, okay? So this substance then goes into the bloodstream and it goes to those active parts that we're interested in looking at, just like you see here. See, and so when the neural activity, which is the neurons, when the activity is high in these particular areas, these color coded areas are going to show on the computer. Okay, so let's say if the neuronal activity in the parietal lobe is what we're interested in. Well, when that's stimulated and when the activity is high, it's going to come up on the screen because we've color coded it so. And of course, what you'll notice that red does indicate the most activity, okay? That's the greatest activity, and it goes in descending order, followed by the yellow and then the green and the blue. Blue typically indicating where there's not a lot of functioning there or activity. So let's say, for instance, if a person was working on a task of recognizing images, okay, and there may be shown a series of cards of animals, and they're being asked to name those animals. Well, while they're naming those animals, those neuroscientists are looking at that particular area of the brain and its activity. So if something was to show up blue hypothetically, then we can see that, okay, where our ability to recognize and recall and label, which is typically verbal memory, may not be working in that area. So those parts of the brain that's active may show us just that based on the color code. Okay? Or for instance, let's say we're shown a series of cards of words and we're required to say those words. And a person may have had some issues or maybe damage in the Broca's area. Remember that? Well, we may see that area may light up a particular color, indicating if there's activity there or not. And behaviorally, we'll see a difference. If they're not able to say the word at all or produce the speech, then it may light up blue, and vice versa. Okay. So with the PET scan, then, it's very great in capturing these particular types of activities, and so although it's a very great imaging technique to use, of course, one of the downsides is that it is very expensive. It is very, very expensive. And it does require a sophisticated staff or trained staff to really use it and operate it. So typically your brain surgeons or someone who has specialized in the field of neuropsychology or neuroscience would typically be your ones doing these types of imaging techniques, okay? However, we can kind of say that a test like this is kind of justified for being expensive. I mean, after all, you're able to inject a color into our bloodstream to tell us whether or not certain parts of our brain are still active or not. I mean, you could possibly help someone prevent a stroke that way or even Alzheimer's that way. So it kind of makes sense for a test to be this kind of expensive. The other disadvantage of the PET scan is that it doesn't really detect changes in behavior that are briefer than 30 seconds. So, let's say if the person is looking at the cue card with the word that says dog, and let's say they say it within 5 seconds. Well, the PET scan may not be able to capture that kind of activity compared to if a person took 50 minutes to say the word dog. So that's another disadvantage of it. And it doesn't image the brain tissue itself. So it's just really you're seeing the color, not necessarily the tissue. So that's just two other disadvantages of the PET scan. So we know one major advantage is sophisticated. It does detect brain activity in real time. However, it tends to be very expensive. And it doesn't really capture behavior that's less than 30 seconds. Okay. And as we talked about here, we already said that earlier that the advantage is that it does track changes in real time. It can be very expensive, and it can be very slow at times. So then, what do we do? Is a PET scan the only option we have if we want to look at brain activity? No, it's not. So we have the fMRI. So now we're talking about functioning. If we remember earlier that the MRI by itself allows us to detect the structures, right, like a tumor, through using through radioactive activity to locate that. Well, with the functional MRI, then it measures brain activation by detecting its oxygen levels in the active structure. So your brain does need oxygen. That's just like when I know you probably heard before of situations of individuals who were born with a lack of oxygen to the brain and those types of situations. Your brain needs oxygen, and your brain needs oxygen in order for the structures to work properly. And so, what the fMRI does is that it measures that brain activation of those particular structures based on the level of oxygen that's there. And as we can see here, These are an example. This is an example of individuals that were processing words and being able to recall. So we're looking at, delayed and short recall. Okay. So as we can see here, it's color coded based on the level of oxygen in those areas. So if we kind of could look here, we could kind of see that where there was a lot of forgetfulness was probably here compared to here. So I'm going to stop right here, okay? Because we've covered a lot so far, and just to cover up with the fMRI, the advantage is that it does provide great spatial resolution. So we're talking about location and the image being very clear in order to see that, and it's very timely. And you don't need any kind of radioactive substances injected into your body to do so. However, it does have its disadvantages. It tends to be very expensive and very slow. So I'm going to stop right here. And so for homework, what I want you to do is that I want you to type up the or list all of the brain imaging techniques we've talked about thus far. Okay, that's the CT scan, the EEG, the fMRI, the MRI, the PET scans, and I want you to tell me the advantages and disadvantages of each. Okay. So you're going, and if you want, you're more than welcome to make a table if you want to, and do so. And you're going to submit that to me on Moodle by 11:59 p.m. And I hope that you guys were listening and taking notes, because when we get to class, we're going to kind of put all this back into perspective. Okay? If you have any questions, please feel free to email me. You guys have my email. If not, everything is lowercase. It's B as in boy, C as in cat, my last name, POINTER, and the number 1 at gmail.com.